Today's video is about safety in the biology laboratory. Teachers and students must be aware of the importance of safety precautions associated with microbiological experiments. All work must be supervised by a trained teacher or qualified supervisor. Microorganisms are a potential hazard to persons performing microbiological experiments. Working with microorganisms requires special handling, storage, and disposal techniques. There must be proper use of aseptic handling techniques at all times, such as wearing of gloves and proper washing of hands. Here are some scenarios where accidental laboratory-acquired infections may occur. All cuts on the body surface should be covered with waterproof dressing before starting microbiological experiments. Liquid disinfectants and germicidal agents generally have limited effectiveness. For complete sterilization, all apparatus used in microbiological experiments must be autoclaved. Only known microorganisms from recognized suppliers should be used for inoculation. Microorganisms of unknown pathogenicity or from unknown sources should not be brought into a school laboratory without proper precautions and authorization. For example, attempts to isolate unknowns from soil culture. Those school laboratory experiments are performed using microorganisms that are unlikely to cause disease in humans and animals, there are still safety guidelines to be followed. Here are some of them. The microorganism is cultured in a plastic petri dish or other standard non-breakable container and sealed. A culture must not be opened for identification, subculturing, or isolation. The experiment involves only procedures in which the petri dish remains sealed throughout the experiment, such as when counting the bacterial colonies. In this context, the culture is of a low-risk level, containing low-risk microorganisms. The sealed petri dish must then be disposed of in the appropriate manner under the supervision of the teacher or trained laboratory technician. Students must be reminded to always report any spills involving microorganisms to their teacher. During a microorganism spill, students should stay away from the spillage immediately, and it should be cleaned up by the teacher or laboratory staff. In addition, other laboratory users must be alerted or warned of the spill. Clothing around the affected part should be removed. Hands and the affected part should be washed with soap and water, and a doctor should be consulted immediately. If the spill gets in contact with the eyes, flush the eyes using the eye washers in the laboratory for about 15 minutes. Ophthalmic saline irrigation solution may also be used. Call or consult the doctor for further advice.